Emily Powell. She's a former student in our phage genomics course, so she's going to summarize some of the work from that class and then extensions that she's made on this herself. And I'll just kind of sing some praises for Emily. She has been working for the past semester and a half now at this point in the lab 2 times a week, receiving no official credit for it. So she's committed. That's usually 4 to 5 hours a week. So she's working hard, and she's here to talk about some of the things that she's done over the past year and a half.
This presentation is on the mycobacteriophage discoveries in Cathedral Caverns, which is located in Grant, Alabama. A little introduction. Bacteriophages, which is what I'm going to be talking about, are viruses that can infect and destroy bacteria, which is really good because that's, I guess, a step towards medical research, and we can use these in that. They can be used as antibiotic treatments and everything. The reported study focused on the isolation and characterization of the bacteriophages that infect Mycobacterium sphagnum. That's the host that we use. There are multiple others like Mycobacterium tuberculosis and others. Bacteriophages that target mycobacteria are called mycobacteriophages. My objectives were to isolate the novel mycobacteriophages from a subterranean environment, which in this case was a cave. Determine the structural morphologies of these unique mycobacteriophages via transmission electron microscope analysis, and sequence and annotate the genomes of these isolated mycobacteriophages and compare them to other phages in the database. For capturing, I took at least 4 samples to begin with last year, and the cave, the one that got the results was the sample that I took right here on this bank, which is located next to Mystery River, which runs naturally through Cathedral Caverns. You can see in very far off zoom of it and then up close. My materials, we did soil samples that were enriched for the isolation of the phages, which we used Mycobacterium sphagnum as a host organism. We identified potential plaques or zones of clearing that were caused by viral infections on the lawns of the Mycobacterium sphagnum. And the plaques were then purified by multiple rounds of plating until consistent morphologies were observed. What we mean by lawns is if you can see the cloudy areas right here and then the clear plaques also right here. And also, when we have to do multiple rounds of purification is because this virus that I'm talking about right here has 2 distinct morphologies. These little clear bullet holes that we call them, and then the larger halos that you can see. We had to do multiple rounds of purification because we thought that there were 2 different viruses on the same plate, but we finally got it down to 1. Continuing, the DNA was isolated from the purified bacteriophage and then was sent off to be sequenced. The genome was then annotated. We used DNA master software, which was really interesting. Then we went down to UAB and were able to use their transmission electron microscope, which is a really cool thing. We used it to visualize the viral morphology, and then the genomic and morphological data was then entered into an international bacteriophage database. The results were that the isolated plaque with a halo-like morphology, which you can see right up there. The phage we named CaptainTrips after the Stephen King virus in the stand. The plaque diameters were about 1 millimeter after a 24-hour incubation period. And the restriction enzyme digestion analysis revealed that CaptainTrips was very unique to the other bacteriophages that were found in that lab. So that's why it was chosen to be sequenced. Here you can see a picture from the transmission electron microscope that we used. And you can see the virus. Bacteriophages usually have a capsule or capsid as their head, and then a tail And you can't really see them right here, but they have spindly little fibers that kind of look like spider legs to attach to bacteria, and then they inject their host with their DNA material. It was a non-enveloped head, and it had a non-contractile tail. And it belongs to the Cyphobiridae family of bacteriophages. CaptainTrips, the genome size was about 57,000 base pairs, which is normal for mycobacteriophages. We annotated 105 genes in this, and you can see the top picture up here is the full DNA that was sequenced using DNA master. It belongs to the mycobacteriophage cluster F, and cluster F is relatively rare. It represents about 10% of the entire phages that have been discovered, and they range from A to S. This picture right here is of the phage database that I use. It's called phagesdb.org. You can get on here and look for just different phages. It's really interesting to see all the different morphologies and the locations. This is the only reported mycobacteriophage to be found in a subterranean environment, which in this case was a cave. It's the member of the mycobacteriophage cluster F, which I said represents about 10% of all the genomes. And it contains genes that suggest that CaptainTrips might have a host range beyond any of the mycobacterium. We found one gene, which is circled right here, that actually is a lot like another gene that we found in a Streptomyces phage, which you can see that at the top. Continuing research, I've isolated 4 additional mycobacteriophages from the same area in the cave that I took my samples from, and using a culture-dependent methodologies to assess microbial diversity of soil samples in an attempt to identify potential hosts for mycobacteriophages we have discovered. We also have used the culture-independent techniques, DNA isolation, and PCR analysis. Acknowledgements. I'd like to thank Howard Hughes Medical Institute, the JSU Department of Biology, Dr. Murdoch and Dr. Debro, and Dr. Blair for his assistance with our bacteria.